everyone. My name is Alyson Lang, and my presentation is about transgenic plants and science communication. So start off with who am I? I'm Alyson Lang, and I'm a sophomore at Eastside Preparatory School around Seattle, Washington. My mentor was Matthew S., and together we made this project called The Process of Plant Transformation. So I'll go over the big picture of why I chose this project, my interest in the future of plant biotechnology, the process of creating transgenic plants, which is the bulk of what my project is about, and lastly, science communication. And so the big picture is this project comes in the form of a personal blog on blogging platform Medium because I thought this would be the most accessible way to disseminate knowledge. And in line with that, my goals were to firstly contribute to science communication and secondly to explore plant and microbial biotechnology. And so through this blog, I have so far published 6 different blogs. 4 of them are in this group that explains the process of creating transgenic plants. We'll go over each of these 4 steps later. All right, now the future of plant biotechnology. Why am I interested in this field? Well, this is a pretty niche and small field. Not many people know of it. A lot of people just think, oh, plants, they're just there. But with the onset of like gene editing technologies like CRISPR and the explosion of biotech in general, there's a ton of different fields that this science can contribute to. For example, nutrition, carbon removal, heat and drought resistance, food production efficiency and cost, air purification, even home decor and space travel. So I just feel like the possibilities are limitless here, but it is an underexplored and undervalued field. And of course, with every new technology, we must advance with caution and communication, which is why I here am attempting to try to spread the knowledge of this technology to the public, and so that it's not only reserved for those mysterious high-up scientists. Now the process of creating transgenic plants. I'll quickly go over the subjects of my 4 different blogs. The first step is called artificial gene synthesis. So the end goal of this step is to put the genes of one organism into a plant, making that plant a transgenic plant. So how do you get that gene in the first place? So there's 2 techniques that are usually used. First is to buy one from companies which can use this pretty complicated process to synthesize small segments of DNA from scratch, and then use those small segments to isolate larger segments. And the second option is to do that yourself and using technology called like PCR to take a small section of a gene from a larger construct called a plasmid, which is the purple circle you see on the right. And many genes must be put together to make a functional plasmid because there are many genes, all of which have a different part in a specific method that is necessary to, put the genes that you want into the plant, which we'll talk about later. And the desired gene you can see is like the cyan, tan, teal, bluish part on the right. And through processes like Gibson assembly, Golden Gate cloning, and yeast assembly, which are all different methods of molecular cloning or isolating that blue part from the purple, we can get many small segments of blue that we then put together into a whole new plasmid. All right, next is gene insertion. So unlike human or animal cells, plant cells have a pretty thick cell wall, and it is pretty difficult to penetrate. There are some techniques which include protoplast transformation, that is basically dissolving the entire cell wall, but unfortunately that makes the cell extremely easy to break and pop and die. So not very popular method. And number 2 is biolistics, which sounds like ballistics, and it really is. So you basically shoot a little pellet of metal covered with DNA into plant leaves, and that spreads the DNA into the cells, but it's not as efficient or effective. Third is the most popular method, which is agroinfiltration. This is the use of Agrobacterium tumefaciens, which is a plant pathogen with an innate natural ability to inject DNA into host plants and creating tumors. So the image on the right is the effect of wild-type Agrobacterium and tumefaciens infecting a plant. But in a lab, we do not want those tumors to grow. So the Bacteria that scientists use usually are edited already to take out some genes that creates the tumors and to generally make them more suitable for gene insertion. Third is micropropagation. So in order to make a plant transgenic, every single cell of the entire plant must be transgenic. And that's really, really hard to do if you just try to cover a plant in agrobacteria, because not all of the agrobacteria makes it. So in order to make an entire plant transgenic, you need to start from a small portion. And the first picture you see are just cut up leaves of a plant. And these are infected by agrobacteria and then put put onto an agar plate with some hormones and nutrition that prompts it to grow into the second picture, which are called calli. Calli are basically just clumps of stem cells. And in the first picture, those cells were specialized leaf cells, but um, after becoming calli, they revert back into a stem cell form, which is extremely interesting. This experiment that you see on the right is a scientist trying to put a ruby gene, which makes the plant red, into this plant. So you can see on the calli stage that only a couple of the calli becomes red. So then from those few, we like isolate them and put them into a different hormone nutrition mix, which prompts the calli to grow back into a full plant, like you see in the third picture. That's really interesting. So yeah. All right, Fat. So what is the impact of this technology and my project? So first I want to talk about science communication and why that's a big deal. So first, there's a pretty big gap between scientists and the public, despite many subjects being of equal relevance to all of humanity. For example, space exploration, climate change, food safety, etc., that affects all of us. Yet, well, the public only has a small portion of the knowledge that maybe scientists would like so that they can support each other. And the risks of this is destructive science, political polarization, misinformation, division, and like a general impediment of progress. Because if the public like views the scientists as mysterious, and then that might lead to distrust and lack of support and suspicion. And I know that it's impossible to like make the public understand everything, but there are a lot of misunderstandings in the past. For example, the huge GMO debate and ongoing debates such as those. So I hope to make the playing field a little more even. So my project in science communication. I would like to educate and inform the public in an understandable and engaging way as a companion, not as a teacher, which is why I chose to do this project in the form of a blog post on Medium instead of a research project or anything else that maybe would have allowed me to explore the project deeper, but would not have made it very accessible to others. Second is to explore relevant biotechnologies and their impact on our future, because I'm genuinely interested in this subject, and I think there's a lot of potential, and it deserves a little more recognition. And third, I would like to offer opinions, spark curiosity, and encourage interaction, because for me, just something as simple as watching a Netflix show or reading an interesting article, like really sparked my interest, and I hope that maybe a little more exposure could do the same for others. All right, thank you. Any questions?
Uh, Thank you, Alyson, for the topic. I will say that you succeeded in your goal because when you first were talking about it, I'm like, oh, this is really interesting technology. I could see how people might be a little bit worried about it. And then you educated. And I think that one of the things that was really impactful was the visual. Like when you got to the visual screen of the three separated images, and you could see how by developing these plants, you could turn this little leaf into actually this red plant at the end, was pretty remarkable. Why do you think, I mean, because it does seem like it was a pretty image to see. What do you think, what do you think are some of the biggest benefits and the biggest risks with doing something like this?
Yes, so I can start with the benefits, I guess. Well, you could technically put any gene into specific plants, and not even just plants, like bacteria or other microorganisms too. So we could basically change the genetics of life, which is dangerous, but also there's so many possibilities and so many advances in science that we could accomplish if we were to do this safely. One of my other interests is like biotechnology and space exploration. So I guess I could explain a little bit on that. So astronauts in space need food, and on Earth, food in the end comes down to plants. So there's a lot of work going on at the moment trying to make plants that would be more easy to like I don't know keep alive in space, and maybe in the far future contribute to like terraformation of Mars or exciting stuff like that. And in a more I don't know, applicable sense and of right now, climate change is a problem and it is affecting the natural world a ton, and plants obviously make up like what 80% of the biomass on Earth. So if we could just alter a few of those plants to maybe especially crops to survive on less water or in hotter conditions, that could prevent famine in a lot of countries. And after these plants are developed in a scientific lab and gone through all the safety procedures, they can be easily like disseminated into the poorer countries and the people who need them. So it's a technology that could really change the world. And the risks, obviously, well, where do I start? But if scientists just like carelessly insert genes into plants or microorganisms without knowledge of complete knowledge of its effect and impact, then like it could possibly disrupt uh, existing communities, and it's highly unlikely that any GMOs will like poison you or give you cancer or anything like that. But there are some like minor side effects that are also harmful. But yeah, I do advocate for the exploration of this technology, but I recognize that there is a lot that needs to be done in order to make sure that it is completely safe.